It's my great pleasure today to introduce Dr. James L. McGaugh. He's a distinguished professor emeritus in the Department of Neurobiology and Behavior, which is my department at University of California, Irvine. I will note that when he started in this department, pushing 60 years ago now, the department didn't exist at the time. He was the first generation of faculty here at UC Irvine and really played an instrumental role in the history of this university in many ways. In fact, I'm currently speaking to you from McGaugh Hall. I'm going to talk to you today about, Dr. McGaugh is going to talk a little bit about his career today. And he's going to mention, he published 15 times in the journal Psychopharmacology. A lot of his most important findings have had to do with pharmacology research. And it's really just a great honor to speak with you today, Dr. McGaugh. Welcome.
Thank you very much. Pleasure to be here.
Great. Well, maybe we could start by you telling us a little bit about sort of the trajectory of your career. Did you always know you were going to be a scientist, for example?
No, in high school, I was interested primarily in drama and music. I acted and I played in the concert band and the orchestra and also in choral groups. And in physics and chemistry, I saw those just as dull things that had to be done to get into college. You know, there was nothing exciting about them at all. So I went to college as a drama major with a music minor. And along the way, I got transformed. I took some courses that looked exciting. I tried a lot of different sciences, by the way. And genetics came close, but then I got into experimental psychology and I found a home there. I really found a home and I changed my major. And that was the period of enlightenment for me when I was a junior in college. And when I went to graduate school, it was all set at that time.
Wow. So when you went to graduate school, then you had an idea that you were interested in experimental psychology, as well as obviously your other kind of more cultural hobbies. What did you do then? Did you just kind of find a lab that did that and plug in and learn everything, or how did it go?
The Graduate School of Berkeley was a very free-form graduate program where you could do almost anything you wanted to, but at the end you had to pass exams. So the exams are sitting there at the end of 5 or 6 years or whatever, and you took whatever courses you wanted to and passed the exams, hopefully. But I got an opportunity to work with a group that was just being formed, looking at the involvement of the cortex in learning, and particularly not only anatomically, but they were interested in acetylcholinesterase or acetylcholine. And so this is a group that was looking at the relationship between acetylcholine and behavior, and I joined that group. And I did, actually my thesis was primarily on that topic, where I'd train animals, and then later on I would sacrifice them and take out the regions of the brain and analyze for acetylcholinesterase. And the assumption was the more acetylcholinesterase there was, the less acetylcholine there would be, because that's the enzyme that hydrolyzes it. And I was looking for a relationship between then that enzyme and the previous learning. And in the course of that, it occurred, it's a very boring kind of experiment. It takes a long time and it's very careful, and a long time in a white lab coat sitting there analyzing. And I thought, wouldn't it be a lot better in place of measuring, why couldn't I manipulate? And so I went to the library. In those days, you had to go to the library because there was no PubMed. And I found a journal, and it said that there was a previous experiment that showed that strychnine sulfate was an acetylcholinesterase inhibitor. Maybe a mild one, but maybe it was. So I decided to look into that, and I discovered that there had been one previous experiment in the literature showing that strychnine given to animals before they were trained in a maze, their performance was improved. And so in my first experiment, I replicated that, and I found, yes, this drug given each day before training improved their learning. That is, they learn faster. And then I thought, I need some controls here. How many controls do I need in order to exclude for the performance effects of the drug? and maybe 16 or 17 or 18 when you think about it. You have to control.
So you mean basically because the drug, for example, could make them run faster or something like that?
Run faster or slower or hungrier or thirstier or, lots of things like that. So I had read about actually not too much earlier, just a few years earlier, experiments with electroconvulsive shock showing that if animals were given an electroconvulsive shock after they were trained, they didn't remember very well things that were learned within 15 minutes. And I thought, well, if that's the case, maybe I could enhance the memory of the drugs of the animals by giving the drug after training rather than before. So I went to my advisor and I said, I have a fantastic idea. I'm going to do this. He said, why would you do that? The learning is over. And fortunately, he left town on a sabbatical leave, and I was able to do the experiment, and it worked. So I did, I guess, the first ever study using a post-learning drug administration and found enhancement of memory. And I was very excited. As a matter of fact, I think I floated in the air on that. So I published that, and then my career began at that point.
Wow, that's really exciting. I feel like there's a lot of lessons here. Like sometimes, great ideas are, you just kind of got to try it, and you never know, it might work. And sometimes the people that's supposed to know best, like your advisor, in fact, your instincts were better than his.
Yeah, and it also shows how previous findings that seem to be not closely related when put together make sense. Like I had the retrograde amnesia studies, I had the prior study of the strychnine effect injected beforehand. And by that time, I had a lot of experience in training animals. I mean, I did that every day, Christmas, New Year's, and everything. I was doing that. So it all came together. And the miracle for me was that it worked. And that really launched my career. That finding started my career.
Yeah, it's a really very, I think, still commonly used type of manipulation to really prove that whatever you're doing has to do with the memory in particular, as opposed to, any kind of performance. Does the animal actually experience the thing as much as stuff? Just give the drug after the session. Can I ask you, how does this, so I assume that the mechanism here has to do with the idea of consolidation of the memory, that there's sort of an active process that's occurring after the actual learning occurs. Yeah. That is subject to manipulation by pharmacology, for example. Could you speak a little bit to that connection?
Well, the first experiments that we did were simply designed to confirm and extend. And the question was this something that was unique to this particular stimulant drug? So we did a lot of experiments with a whole variety of stimulant drugs, and they all have the same effect, including amphetamine, which is important. And so at one point, we decided then that we really ought to be looking more at mechanism now that we had good descriptive findings. That is, we nailed down the post-consolidation, the consolidation effect. We presumed that it was doing something to enhance the processes underlying memory. And we took advantage of the amphetamine effect because we got the idea that perhaps catecholamines were involved. Now, this was a period of time in history when catecholamines were very active in thinking. If you think back to the 60s and the 70s, a lot of catecholamine hypothesis research was going on. And we found that amphetamine was very effective. And so that then drove us into the nervous system to try to find out how these drugs might be acting inside to do the job that they do. And we took the amphetamine as a key to that to look at adrenergic systems. So the rest of the next, let's say, 10 years or so, the research was really driven by the adrenergic hypothesis in the brain.
Interesting. And this is, of course, just to sort of remind or tell the listeners here, dopamine was sort of arguably discovered in like around this time, like the late 50s or so. I mean, so this is maybe slightly before what this area you're referring to, but it's almost, this is like this issue of neurotransmitters in neuroscience is in some ways really rapidly developing into our modern conceptualization, I think, at the same period. Is that fair to say?
Yeah, a lot of changes were taking place very quickly. I mean, if you go back before 1950, what could you say? I mean, very little, if anything. And so I started my work by coincidence, right at the beginning, really, of the pharmacological explosion that took place in the 60s, 70s, and 80s. And my early research paralleled that in many respects because we took advantage of the pharmacological findings and applied to the research on memory. So that was a very exciting era, and it was one in which, as a former drama major, I had to spend a lot of time in the library learning about the neurobiology and about the pharmacology, and rapidly because I wanted to use it in the research I was doing.
Interesting. So yeah, I think that's also, it's I think an illustrative example of many people's careers in neuroscience is, you sort of come on the scene at a time when things are, dynamic, and by, a little bit of luck, a little bit of good instincts, you really can kind of make it the start of a career for yourself.
Yeah, there was something else. Sputnik went up, and that influenced the federal government to decide that they needed to invest in research. And so all of a sudden, I was when I became a young assistant professor, for the first time, a young person could apply for a research grant. 2 years before, they couldn't because they didn't exist. And so I was just there at the opportune time to get a start with a research grant that then accelerated my research quite a bit. 10 years earlier, and that could not have happened because there was no NIH grant system 10 years before. So it's a very interesting period in the history of American science and neuroscience, that the luck of having a finding and the luck of having finding funding to support that came together for me in the early 1960s.
Wow, that's so interesting. So I had not really ever thought about that portion of in terms of the NIH funding influence on science. I guess before then, it would have been, these sorts of studies would have just been funded by the department or the university. Yeah. Is that right?
Yeah. The department, I was at the Department of Psychology at Berkeley, and they had an animal laboratory. And when I needed animals, I would just go get animals from them, and they bred their own. So we didn't have inbred strains. We had whatever animals were there. And so faculty too, if they were going to do research with animals, they would just go get them. And then the department would provide a few supplies, like they bought my strychnine for me. It must have cost them $10. I have no idea. But the availability of the grants not only meant that supplies could be obtained, and I moved to mice for a while, and I could buy inbred strains of mice to look at genetic differences in the effects of the drugs. And I could also hire assistants to make the work go. So starting in the mid-1960s, my research program was accelerated with the help of NIH, which I greatly appreciated and still do.
Yeah, that's fantastic. So maybe you could tell us then a little bit more about how your career developed after that. So, after this initial seminal finding,
right.
What happened then? Like, what are maybe some of the most exciting things that you feel like you found or that you've seen happen in the history?
There was, at that time in parallel, there was a lot of work going on, anatomical studies, making lesions in different regions of the brain. And a lot of experiments from various laboratories were showing that something happened when you made a lesion of the amygdala, among other structures. But the amygdala looked to be important to us because there was some modest suggestion that it might be involved in adrenergic system in some way. There were some very vague experiments of that kind, or the findings were clear, but the implications were vague. So we decided to focus on the amygdala, initially making lesions of the amygdala to find out whether or not our effects of systemically administered drugs would not be affected or go away. And we found very quickly on that the effects would go away. And so this clearly suggested to us that these systemically administered drugs were influencing memory in some way by acting with either through the amygdala or with influences that came out of the amygdala in either way. So we moved then to experiments in which we administered compounds directly to the amygdala. And we found, interestingly, the same thing. We found, by the way, just as an aside, every stimulant drug that we found injected peripherally also worked when administered directly into the amygdala. And every drug that we administered peripherally, effects were blocked by lesions of the amygdala, and also we found later it was a subregion of the amygdala, the basolateral nucleus of the amygdala turned out to be the key thing. So this pulled it all together, so that we had the peripheral drugs, and we guessed that the amygdala was somehow involved from the literature. And yes, we found out that the effects of these peripherally administered drugs were blocked by lesions of the amygdala, or let's say removal of it, and also that we could inject them directly into the amygdala and get the same effects. So that shifted our interest then to the basolateral amygdala and its role in influencing memory, which we call it the modulating influence, because our evidence suggested that the action was not within the basolateral amygdala, but rather by activating it, it was the projections to other regions of the brain were important. For example, if you found, when I say we, I mean all of my graduate students and postdocs. I take the credit for it, but I had an army of really very nice students. Found that if you manipulate the amygdala, for example, in a water maze, and it's simply swim to a visible chamber, a platform, that the amygdala will influence that, and it also influence learning to a submerged platform. So it will do either of those things. So you can influence them both. And that suggests then that the amygdala is a non-specific modulator of different forms of learning. So then we investigated that, and I think it was 12 different kinds of tasks. And we found in every single one, the amygdala was a modulator. It does not care what is being learned. It will make a stronger memory of that. And by the way, in our experiments, we would test it and then remove the amygdala, and the enhanced memory is still there. So if we stimulate the amygdala and we get an improved learning, then remove the amygdala and test the animal, the enhanced memory is still there, saying, yes, the enhancement is due to projections to other brain regions, not to changes within the amygdala. So this is the way that the story evolved.
And that's specifically during the kind of the encoding of that memory or the consolidation of that memory.
Yes.
That's right?
Yeah.
And so I guess then, is the amygdala the only structure that you're interested in over the course of your career, or is it your favorite brain region? Do you have another one?
Well, it's just my favorite one. We had to look at the caudate nucleus. We did a lot of experiments with that, showing that the modulation effects there was specific to response learning. I did several experiments on that, and we looked at regions of the hippocampus, and the spatial learning was, of course, restricted. The hippocampal, I'm sorry, the spatial learning was specifically related to the hippocampal region of the brain that was activated. So we did a number of experiments of that kind, and it led to ongoing experiments concurrently by former students of mine that are now investigating in more detail the specific regions that are involved in amygdala activation, depending on the type of learning that's involved. For example, if it's just object recognition, then that's a different region. It's not the hippocampus, or it's not the caudate, it's some other region, and so on. And that's going on, it's multiplying in its effect. So this simple little experiment of the amygdala influencing learning is turning out to be important in understanding what regions of the brain are involved in different forms of learning and how they are influenced by the activation. So it's now an ongoing, not by me anymore, but by my former students in various laboratories throughout the world.
Very interesting. And would you say that the amygdala's role is, to do with all kinds, absolutely all kinds of learning, or what about ones that are completely non-emotional, for example, like just incidental learning or that kind of thing?
All kinds, all kinds.
Okay.
It's involved in just simple object recognition. It's involved in object and context recognition. So animals are trained, an object is in a particular context and animals have to remember that. Amygdala enhances that. It enhances memory of responses that are made. We didn't find any task in which the amygdala failed to modulate. So there are other roles that the amygdala plays. Obviously, different regions of the amygdala do different things. But as far as the basal lateral nucleus is concerned, I'm confident in telling you that I know of no evidence to the contrary, that at least its role is to modulate the consolidation of memories recently formed, and it doesn't matter what kind of memories have been formed. Within 10 or 12 type of experiments we've done.
Interesting. Do you think that the fact that I think all those memory tasks that you mentioned, they all involve basically an animal going over and checking out something that's interesting, maybe because it's new, maybe because it's in the wrong place or in the wrong, the wrong object or whatever. Do you think that sort of like salience has anything to do with the amygdala's role in memory, or is it just simply to do with absolutely anything you learn, even if it's the most boring or trivial thing, period? It's right in there.
We haven't done an experiment where the animals are sitting there and present them with, as you would with a human subject in an fMRI, for example. We haven't done that. But my guess is it would. And I'd also say the more activation of the amygdala, the more evidence you'll obtain. So if it's a very bland test, the blander it is, the less likely the amygdala is to be involved unless you activate it. Now, in our experiments, what we do is we take a very bland experiment. Why should an animal remember that there was a ping pong ball in this box and a square in the other one? This is a mouse or a rat going into a room looking at these things. All right, now, why is that important? Well, I guess it's important to the life of the rat, but we can make it very important by giving a post-training injection. All of a sudden I say the next day, I don't know why it is, but I remember that there was a ball in that region of the container. I have no idea because there's no food there, you see. But you see, underlying this is the overall hypothesis that when you get activated, you're likely to remember better. When the brain is activated, you're going to remember better. And so what we do is make the animal artificially remember better by turning on this particular region of the brain or giving a drug that turns on that particular region of the brain. And it's the way it works. It didn't have to work that way. And I didn't have to find in my experiments that it worked that way. So the conclusion that I'm offering are driven by the findings that we obtained.
Yeah, that's how this is done. That's so interesting. So another thing that you've been, I think, well known for in recent years is your study of people with highly superior autobiographical memory, or HSAM. And you've been on 60 Minutes and things talking about this kind of thing. So just how does that, first of all, like how do you get to study these particular populations? Maybe tell us something about these people. And how did you start to study them after your career of sort of like animal behavioral neuroscience, circuits and structures and drugs? How did you get from here to that particular topic? And what did you sort of learn from your animal work that taught you about what's going on in these people?
I was sitting at my desk one day and I turned on this is in 2000, year 2000, I turned on my computer and there was a message from a person who said that they had a memory problem and would like to meet with me. And I responded by saying, you've got a memory problem, I can recommend you to a clinic. And she wrote back and said, oh no, that's not the problem. The problem is I remember everything. and I'd like to meet with you. Well, I've worked all my life on enhanced memory, so I thought maybe there's no harm in meeting this person. So she came on a Saturday in the spring of 2000, and I didn't really prepare, but I'd been given a large book for Christmas time. Remember, this is right at the turn of the century. So I had a book that was given to me, which was a day-by-day newspaper clippings for the last century. And so I just opened it up. She said that it's this ability of hers to remember a lot started when she was about 13. So I flipped back appropriately and I began to test her about what was the date of this plane crash and when did this movie star die and who starred in this film. So I'm looking at the clippings and she was errorless for that kind of testing. These are big things though, a plane crash and a movie and so on. And so then we devised more tests for her that were personal tests. Unfortunately, she kept a diary. And so we could make tests made based on her diary. And she was also virtually errorless on her diary going back a number of years. At one point, I asked her the date of the last 20 Easters. And she got one wrong, which she quickly corrected, and she's Jewish. So why should she remember those anyway? So we decided to spend some time with her and we put her through a lot of tests. See, this is completely unrelated to the animal work, to the animal memory work, but I thought somehow they might converge because you're strong memory. So we tested her and we found that she was not very good at other kinds of learning. In the laboratory, just learning material, she was just right average compared with everybody. She had this very specialized skill of highly superior autobiographical memory. So that slipped out, that finding slipped out somehow, and it got into a newspaper. And then, to make a long story short, we were on 60 Minutes in 2010. And that got a flood of people who have the ability. So then we were finally able to test a large number of subjects, probably 60 or so. There are many more, but we found there were 60 people of the people who contacted us who really had the ability. And we did a lot of work with them and we verified that they have the ability. We characterized it. We found that they are obsessive compulsive. We found that there are some structural regions of the brain that look a little different, but nothing popped out. And that's it. And we did not find a relationship with the amygdala, as I thought
I was gonna ask, yeah.
that we might find. And so that turned out just to be another avenue. What we basically did was to validate the notion that there is this ability. It's rare, but we found it easily in at least 100, and I still get 2 or 3 a week. People contacting me saying, I have it and would like to be tested and so on. So there are probably hundreds of such people in the United States, not thousands, hundreds, because it is rare. But they do have a lot in common, with obsessive compulsiveness and the strong memory ability, but it does not relate to that. In subsequent collaboration with a group in Rome that I'm working with, we did find some, using fMRI, some pretty strong evidence of an increased activation coming from the hippocampus up to the frontal regions of the brain in the subjects who have this ability. So there are some good things happening with it at the present time, but it does not link closely to the prior work that I did. Sadly.
Interesting. Sometimes these little seemingly tangent things, can be very fruitful, even if they don't end up kind of connecting back to.
Well, I'm not unhappy that we pursued it. I'm happy that we pursued it. I just wish that it had eventually connected more with the other research, but that's the way it is,
Yeah.
You can't make it up.
Yeah, you kind of follow where the data brings you, of course. And out of curiosity, so that connection with the fact that they have obsessive compulsive disorder, I just think really interesting. Do you do you think of these people? Obviously, they're called highly superior autobiographical memories. That says that they have very good memory of a specific type.
Yeah.
But do you think that it's really that they are just better at making memories than normal people, or is it that they are sort of somehow choosing consciously or unconsciously to, file away these particular facts about what happened on certain days and that kind of thing? What do you think is going on there?
Well, it's hard to say. It is tied to their personal experiences of a adventitious kind, and it starts early. So that we have never tested them specifically, but I've had a lot of parents tell me by a lot, 5 or 6 parents independently telling me that they're riding along in a car and their 4-year-old is in the back seat. And the 4-year-old says, "Oh, yes. Oh, I see that restaurant we just drove by. We ate there last November 3rd. It was a Monday." And the hair goes up on the back of the parent saying, "What have we got in the back seat?" Well, that's how it begins, because these people just report this kind of information. And you'd like to think it's important, but we know it's not, because our first subject remembering the dates of all of the Easters. And another one, I asked, I'm looking at my big book, and I said, "Did you know who Bing Crosby was?" And she says, "Yes." Well, how many people today who knew Bing Crosby was? A crooner, a singer in the 1940s. And I said, "You know what happened to him?" And she said, "Oh, yes, he died on a golf course in Spain." And she gave me the date and the day. And I said, "Well, how did you know that?" "Oh, because my mother was taking me to my soccer game and it came on over the radio." Okay, explain that to me. This kind of information is retained, even though it is not of great personal interest to her, as the date of Easter could not have been very interesting to her. And yet, when we bring her into the laboratory and we say, "Please remember these words, it's very important," she doesn't do any better than you or I would under those circumstances. So there's something that is very special about this kind of ability. And certainly it speaks to a very strong retrieval system. There's no doubt about that. They've got to have a system in there which will pick this stuff out quickly and accurately, but it is selective with respect to the experiences that they have, and I cannot explain why. Let me give you another example, and this is on the 60-minute program. There's a woman named Louise Owen in New York who's really very strong in this ability. I asked her to tell me the days on which it rained in New York in 1990, I think I asked her. And she did accurately. Now, she didn't do it quickly. She had to think about it for a second, but she did it. Now, why would she remember the days in which it remained in New York? I don't know. So it is a phenomenon. It is there. You cannot reject it. It is there, and it's got to be telling us something about the organization of the brain, and I think something important. And I hope that this group in Italy and perhaps others will do more investigation to find out what it is. It's also moving in the direction of sort of changing my thinking about what's important. All my academic life, I worked on getting memory in. How do you get strong memory in so it's preserved? And now I think we have neglected to considerable extent the issue of how we get it out. I think that's incredibly important and insufficiently studied. For example, if I ask you now, who was the first president of the United States? You weren't thinking about it, but it'll just jump right out at you. How did that work? How did that happen? What goes on that enables that? Not just where is it stored, but what is the process that goes on? How does the brain do that, detecting and pull it out? So I think that's the future. That if I were not retired, that is now what I would work on.
Is kind of how does the brain select or go and there somewhere somehow? How did it go in there and just pull them automatically, or not be able to pull them, or kind of almost pull them on the tip of the tongue kind of phenomenon?
Yeah. And I think incredibly important, interesting, and clearly worthwhile investigating. And that to me would be the new frontier. Because if you do that, then it's going to tell you more about how memories are stored. You see, so it's a good access into that. Look at that. You can retrieve this. What kind of information is easily retrieved? What kind is hard to retrieve? In the lives of individuals, what are the kinds of things that are easy and hard and so on? And I just think that's a horrendously interesting question for normals, not just for the special people, for all. How do we do it? How do we do it?
Yeah, and then even in some cases, people say, for example, post-traumatic stress disorder has to do with like not being able to not do it.
Yeah.
It's just like the memory just intrudes.
They're there. They're intrude. They're too strong. They're always there. And why is that? And is there something you can do about it? Not from an emotional point of view, but just from a restructuring point of view. How do you restructure so that that doesn't happen? So I really strongly believe that that's a frontier, really a frontier.
Wow. Well, it really goes to show that even in an almost 60-year career in neuroscience, there's still questions left to be asked.
Oh my gosh, we did a little bit like that, a little tiny bit, but I'm happy with what we did. I'm not ashamed of it.
Well, let me tell you that I'll speak for, I think, the entire field, at least of behavioral neuroscience, that we're glad you did that that career, and you've taught us just a ton of things. You're obviously a legend of the field. It's just a key person in the history of behavioral neuroscience, and obviously for me personally, the history of University of California, Irvine. So I'll thank you on behalf of our listenership here for that, and of course also for speaking with us here today for this Psychopharm Friday interview series.
Thank you. Nice to be here.
Thank you so much, Dr. McGaugh.
Okay, take care.